All right, so today what we're going to be doing is we're going to be working through this protein synthesis lab. And the beginning is just kind of a review, so of what we've covered so far with regards to DNA, RNA, and transcription and translation. So please read through it and make sure that you understand it. So the first part is about transcription. And this is when, obviously, we turn the DNA into mRNA. So you're in number one, you are given a strand of DNA, and then on the line, you're going to write the complementary strand of mRNA. So you're going to follow your base pairing rule as outlined above, and you're going to just write that complementary strand there. Then what we're going to do is we're going to start working with the codon wheel, and this is going to be used pretty much for the rest of the lab. Okay, so on the second part, what we're going to do is we're going to actually read the codons. So remember, codons are three-letter sequences. They're kind of like words that we can translate. So A and B are three-letter sequences, and then you just put the amino acid name there. Now, you can go ahead and abbreviate them using the first three letters of the amino acid. The only one that becomes a problem with is glutamic acid and glutamine, because they're both going to be GLU. So for glutamic acid, we're going to use the GLU, but for glutamine, we are instead going to use GLN. Okay, if you want to write them all the way out, that's fine as well, but those are the only two that create an issue with using the first three letters. Okay, and that's how you can work that. Now for these longer ones, what I suggest you do is you actually go in and put a slash mark in between each three, so that way then you know where the codons end and where they begin. Now the last part, you're going to be working with the manipulative that you were given, the blue strip which represents the messenger RNA, and then the yellow pieces represent the tRNA with the attached amino acids. So what you're going to do is you're going to lay down that strip of the blue, and your job is basically to match up the anticodons to the codons. So find the anticodon that matches up with the codon. Now there will be one short, and there's a reason why, and if you look at what it is, that should tell you exactly why. So you're going to go ahead then in number 7, you're going to write the amino acid sequence. The amino acids are on the yellow, and they're just those three-letter codes. So you just write the three-letter codes and you separate them with a dash. After you're done with that, please make sure that all of those little bits of paper get back into the baggie so that it can be used again and we're not missing any. Then you're going to get into your analysis and interpretation. So this is a strand of DNA, and then you're going to list the mRNA, and then you're going to translate that into the amino acids. Now, with this one confuses people. Basically, the idea here is you take your messenger RNA strand that you did here, and you're going to delete off the first base. So the first codon would be AUA. So what I would suggest that you do is you actually write the new amino acid or the new codon sequence here, the new mRNA, and then put your new sequence of amino acids down below. Okay, now because we're cutting off a codon, that means at the end, or we're cutting off a base, that means the end's only going to have 2 letters. That will not translate into anything, so you're actually going to end up being 1 amino acid short. All right, so for number 3, we're saying, okay, this is what we're going to create in terms of our mRNA strand. Okay, you're going to compare this mRNA strand with the one that you created in number 1 up here. The first thing you're going to do is talk about how many mistakes were made in transcription. Then you're going to translate this, and then you're going to write the amino acid sequence. Okay, and then you're going to compare the amino acids that you just wrote to the amino acids that you did in number 1 and tell me how many differences there were. After that, you've got some analysis and conclusion questions. And if I'm remembering right, there is a mistake on one of these boxes, and I can't remember which one it is, so I will make sure that I touch base with you guys. I want to say it's on this one down here on 4 through 6. There's one of them that has a mistake in it. So I will make sure that it is marked on your lab. Okay? And then all you need to do is answer those questions, and then you are done with the lab and you can turn it in.